Hi, in this talk, I will present our work robust recursive partitioning for heterogeneous treatment effects with uncertainty quantification. Given the dataset from a randomized control trial or observational study, we try to identify subgroups of individuals that have similar covariates and treatment response. We want to identify subgroups to be interpretable. As shown in the left panel, we identify two subgroups that are well separated in terms of some simple thresholds on the covariates x0 and x1. Suppose we estimate the treatment effects using black box machine learning model and cluster the individuals with similar estimated treatment response. As shown in the right panel, we will not identify subgroups that are interpretable. In our subgroup analysis method, we allow for any black box machine learning model to estimate the conditional average treatment effect. Due on accurate machine learning model, we construct interpretable subgroups to understand the effect heterogeneity in the space of patient covariates. Also, we want to quantify the effect uncertainties within and between the subgroups. We want our method to work for data from randomized controlled trials and observational study. The previous subgroup analysis method tried to identify subgroups by maximizing the effect heterogeneity across subgroups. It often relies on a specific CAD estimator decision tree. Here is a toy example. Suppose the treatment effect is sampled from a normal distribution with 0 mean and standard deviation 0.1. Clearly, there's no subgroups, but the previous method may identify subgroups due to the noise in the data. In our method, we try to avoid this type of false discoveries by rejecting to identify any subgroups if the confidence interval of the treatment effects have large overlap across subgroups. We start by constructing a conditional average treatment effect estimators for the entire covariate space. Here we can use any conditional average treatment effect estimators, any machine learning models we want to use. For example, we can estimate the treated outcomes by a regression model and estimate the control outcomes by another regression model. And then we take the difference between the estimated treated outcomes and the estimated control outcome as the conditional average treatment effect estimator. Given the conditional average treatment effect estimator, the CAD estimator, we evaluate the effect homogeneities within subgroups by the concentration of the treatment effects within the subgroup centered around the average treatment effect. Specifically, we obtain the confidence interval of the CAD estimator using conformal prediction method. The confidence interval constructed by the conformal prediction specify the frequencies coverage guarantees in finite samples. That is, given any coverage rate we want to achieve, 1 minus alpha. So let's say it's 90% or 95%. The confidence intervals constructed by conformal predictions is guaranteed to satisfy that coverage rates in testing time. Now we develop a confidence criterion for partitioning the covariate space into subgroups recursively. In the criterion, we minimize the absolute deviation of the CAD confidence interval from the group mean. We also minimize the expected confidence interval width because a narrower confidence intervals can also help to reduce the deviation. As shown in the figure, the criterion splits the entire covariate space into two subgroups. The criterions improve both homogeneities within subgroups and heterogeneities across subgroups. After splittings, we reconstruct the confidence intervals using samples within each subgroup. Then the confidence interval satisfies the coverage guarantees within each subgroup. The theoretical guarantees of the confidence interval in our method can help to avoid false discoveries of subgroups. If the confidence intervals have large overlap across the constructed subgroups, the constructed subgroups are not well identified. Conversely, if the confidence intervals have little and no overlap across subgroups, the subgroups are well identified. Finally, we evaluate our method R2P by comparing its performance with the existing subgroup analysis method. For this, we mainly use the variance across and within subgroups to evaluate the heterogeneities and homogeneity respectively. From the tables, we can see our method achieves the best performances, meaning larger heterogeneities across subgroups and smaller variance, larger homogeneities within subgroups. Also, the confidence intervals of our methods are much narrower than the competing methods while satisfying the coverage guarantees for each subgroup. In the figures, we compare the treatment effects at subgroups level for all the methods. Our method R2P discovers the subgroups with minimum overlap to avoid false discoveries. To sum up, in this works, we developed a robust recursive partitioning method for subgroup analysis. Each subgroup discovered by our method is interpretable based on the tree structures in the recursive partitioning process. Our method can be used with any black box machine learning models and enable them to produce trustworthy and interpretable treatment effect estimates in practice. Thank you for watching. If you're interested, take a look at our papers and softwares on our website.